Alright, hi guys, my name's Julian, and this is Jerry. We're going to be bringing you 5.4, Modification of Cell Surfaces. Extracellular structures take shape from materials the cell produces and secretes across its plasma membrane. In plants, prokaryotes, fungi, and most algae, the extracellular component of the cell is a fairly rigid cell wall. A cell wall occurs in organisms that have a rather inactive lifestyle. Animals that have an active way of life have a more varied extracellular anatomy appropriate to the particular tissue type. Cell surfaces in animals. We will consider two different types of animal cell surface features. One, the extracellular matrix, short for ECM, that is observed outside cells, and two, junctions that occur between some types of cells. Both of these can connect to the cytoskeleton and contribute to communication between cells and therefore tissue formation. Alright, hey guys, this is a 5.4 student summary with my self-made constant map. I'm Julian and this is Jerry. Alright, so the extracellular matrix is what we learned about in 5.4, which is basically the all around the cell membrane, or the membrane of the cell. So all the plasma juices all around the cell that connect cells to other cells, that's what the extracellular matrix is. And in animals, the way the walls connect to other walls of different cells are through desmosomes, and they connect in three different ways: with an adhesion junction, a tight junction, or gap junction. And the difference between them is that the adhesion junction attaches with filaments. The tight junction is tighter, as it says in the title, and it attaches via proteins. The gap junction also attaches, but instead it does it with its plasma membrane, and it allows communication between the cells. And for our plants, you have the cell wall instead of the plasma membrane, and the way they connect to each other is through the plasmodesmata, and that's attachment via the cell wall membrane. Alright, thank you. That's all for 5.4. Alright, extracellular matrix. A protective extracellular matrix is a meshwork of proteins and polysaccharides in close association with the cell that produced them. And this is the polysaccharides inside of the cell that it was produced in. Collagen and elastin fibers are two well-known structural proteins in the ECM. Collagen resists stretching, and elastin gives the ECM resilience. Fibronectin is an adhesive protein colored green in figure 5.13. Here's the fibronectin, a little green horseshoe right here, that binds to a protein in the plasma membrane called integrin. Integrin is this red that we have here in figure 5.13. Notice that the integrin also makes contact with the cytoskeleton inside the cell. Through its connections with both the ECM and the cytoskeleton, integrin plays a role in cell signaling, permitting the ECM to influence the activities of the cytoskeleton and therefore the shape and activities of the cell. Amino sugars in the ECM form multiple polysaccharides that attach to a protein and are, therefore, called proteoglycans. Proteoglycans, in turn, attach to a very long, centrally placed polysaccharide. The entire structure, which looks like an enormous bottle brush, resists compression of the extracellular matrix. Proteoglycans assist cell signaling when they regulate the passage of molecules through the ECM to the plasma membrane, where receptors are located. During development, they help bring about differentiation by guiding cell migration along collagen fibers to specific locations. In short, the ECM has a dynamic role in all aspects of a cell's behavior. When we study tissues, we will see that the extracellular matrix varies in quantity and in consistency from being quite flexible as in loose connective tissue, semi-flexible as in cartilage, and being rock solid as in bone. The extracellular matrix of bone is hard because in addition to the components mentioned, mineral salts, notably calcium salts, are deposited outside the cell. Junctions between cells. Certain tissues of vertebrate animals are well known to have junctions between their cells that allow them to behave in a coordinated manner. These junctions are of three types shown in figure 5.14. Here's the three types of junctions that we have in the cells of animals. Adhesion junctions, this one, serve to mechanically attach adjacent cells. Two types of adhesion junctions are described here in desmosomes. Internal cytoplasmic plaques firmly attached to the cytoskeleton within each cell are joined by intercellular filaments. The result is a sturdy but flexible sheet of cells. In some organs, such as the heart, stomach, and bladder, where tissues get stretched, desmosomes hold the cells together. At a hemidesmosome, a single point of attachment between adjacent cells connects the cytoskeletons of adjacent cells. Adhesion junctions are the most common type of intercellular junction between skin cells. Adjacent cells are even more closely joined by tight junctions, in which plasma membrane proteins actually attach to each other, producing a zipper-like fastening. The cells of tissues that serve as barriers are held together by tight junctions. In the intestine, the digestive juices stay out of the body, and in the kidneys, the urine stays within the kidney tubules, because the cells are joined by tight junctions. A gap junction, and this one right here, allows cells to communicate. A gap junction is formed when two identical plasma membrane channels join. The channel of each cell is lined by 6 plasma membrane proteins. A gap junction lends strength to the cells, but it also allows small molecules and ions to pass between them. Gap junctions are important in the heart muscle and smooth muscle because they permit a flow of ions that is required for the cells to contract as a unit. Plant cell walls. In addition to a plasma membrane, plant cells are surrounded by a porous cell wall that varies in thickness. Depending on the function of the cell, all plant cells have a primary cell wall. The primary cell wall contains cellulose Plant cell walls. In addition to a plasma membrane, plant cells are surrounded by a porous cell wall that varies in thickness depending on the function of the cell. All plant cells have a primary cell wall. The primary cell wall contains cellulose fibrils, in which microfibrils are held together by non-cellulose substances. Pectins allow the wall stretch when the cell is growing, and non-cellulose polysaccharides harden the wall when the cell is mature. Pectins are especially abundant in the middle lamella, which is a layer of adhesive substances that holds the cells together. Some cells, in woody plants, have a secondary wall that forms inside the primary cell wall. The secondary wall has a greater quantity of cellulose fibrils than the primary wall, and layers of cellulose fibrils are laid down at right angles to one another. Lignin, a substance that adds strength, is a common ingredient of secondary cell walls in woody plants. In a plant, the cytoplasm of living cells is connected by a plasmodesmata. In figure 5.15, we see a plasmodesmata in its model. Numerous narrow membrane. In a plant, the cytoplasm of living cells is connected by a plasmodesmata. In figure 5.15, we see a model of a plasmodesmata. Numerous narrow membrane-lined channels that pass through the cell wall. Cytoplasmic strands within these channels allow direct exchange of some materials between adjacent plant cells and eventually all the cells of a plant. The plasmodesmatas are large enough to allow only water and small solutes to pass freely from cell to cell. This limitation means that plant cells can maintain their own concentrations of larger substances and differentiate into particular cell types. Connecting the concepts of Chapter 5, the plasma membrane is quite appropriately called the gatekeeper of the cell because it maintains the integrity of the cell and stands guard over what enters and leaves. But we have seen that the plasma membrane also does much more than this. Its glycoproteins and glycolipids mark the cells belonging to the organism. Its numerous proteins allow communication between cells and thereby enable tissues to function as a whole. A new endeavor is to understand how the extracellular matrix in animal cells assists the plasma membrane in its varied functions. The progression in our knowledge about the plasma membrane illustrates how science works. The concepts and techniques of science evolve and change, and the knowledge we have today will be amended and expanded by new investigative work. Also, basic science is applications that promote the health of human beings. To know that the plasma membrane is malfunctioning in a person who has diabetes or cystic fibrosis, or in someone who has a high cholesterol count, is a first step toward curing these conditions. Even cancer is sometimes due to receptor proteins that signal the cell to divide even when no growth factor is present. Our ability to understand the functioning of the plasma membrane is dependent on a working knowledge of the molecules that make up the cell. We continue this theme as we discuss metabolism in the next 3 chapters.